Hello everyone, welcome. My name is Jay. Let's get started. Okay, so we're going to keep this webinar fairly high level and introductory. First of all, we want to do a quick overview of GenomeStudio 2.0 in general, but as the title suggests, we will focus mostly on the Polyploid Genotyping Module. We'll learn how to download and install GenomeStudio 2.0. We'll identify differences between the Polyploid Genotyping Module and the standard Genotyping Module. We'll define the two types of polyploidy that are supported in this module, autopolyploidy and allopolyploidy, and we'll understand the use and options for the two different polyploid clustering algorithms, Polygen Train and DBSCAN. So the first thing you should understand about GenomeStudio 2.0 is that it's actually only two modules. It's the genotyping module and the Polyploid genotyping module. The methylation and gene expression modules are still in version 2011.1, which is still available for download on the GenomeStudio support page. While license keys are still required to install the methylation and gene expression modules, they do not have to be purchased. They are actually in a text file with the download for the installer. There's no licensing mechanism for GenomeStudio 2.0. The only thing you have to do is at the very beginning, you have to sign in with your Illumina login credentials. That being said, you do require an internet connection in order to install the software. It's important to realize that GenomeStudio 2.0 is backwards compatible. You can open version 2011.1 projects in 2.0, but not vice versa. Any projects that you create in 2.0 cannot be opened in 2011.1. And lastly, why was this software actually versioned? The primary reason was actually to support our Infinium XT product. But we also did introduce a new revision of our Gen Train algorithm, which includes the normalization and clustering algorithms, and we improved upon the previous polyploid clustering module. So you get GenomeStudio on the GenomeStudio support page. You will notice that the support page does have a new format, and it's actually grouped by application now. So under the genotyping application, you will find the installer for GenomeStudio 2.0, the 2.0 plugins, which were updated specifically for this release, and a sample polyploid genotyping data set. Under the methylation and gene expression applications, you will also find the GenomeStudio version 2011.1 installers, as well as the text file that includes the license keys that you need to install these modules. For those of you that may be aware, we did have a previous polyploid clustering module. That has been archived. If you happen to need it for any reason, we can get it by request. So in order to download it, as we said, just go under the genotyping application, click on the GenomeStudio 2.0 link, and then click on the link for the installer. Once you download the installer, simply run it, and you'll see this screen populate in front of you. This is where you need to input your Illumina credentials, and you only need to do this once. After that, the software will be authenticated, and you can use it without any further login. If you do all this successfully, this is what you should see for your GenomeStudio 2.0 homepage. Note that we only have the polyploid genotyping and genotyping modules. So now that we've reviewed 2.0 in general, let's dive right into the polyploid genotyping module. First, a brief background on polyploid clustering in GenomeStudio. This is actually not the first time we've offered this, but we do consider this to be the most complete solution. We actually introduced manual definition of up to 5 clusters per loci in 2010.3, and then that was carried over into 2011.1. And then in 2012, we actually introduced a standalone polyploid clustering module. But this didn't actually generate genotypes for you. All it did was assign samples to arbitrary cluster names, which wasn't terribly useful for our customers. So now we have GenomeStudio 2.0. It's a real polyploid genotyping module, and it allows you to actually generate genotyping data for your polyploid species. There are a few key differences between the polyploid and diploid analysis in GenomeStudio. On the top level here, you see our general Infinium workflow for analyzing Infinium data. You'll notice that the key differences are when you create the project in which you have to define the ploidy, and note that we only support ploidies of 4, 6, or 8, and this is where you also define the type of polyploidy, which we'll get into in the next slide. The next major difference is actually in clustering the Snips. Instead of just using our standard Gen-Train algorithm, you will use either the Polygen Train algorithm or DBSCAN. And finally, the next big difference is in exporting the data. When you export the polyploid cluster files for use in future products, they will have a different file extension than our usual genotyping cluster files, which are .egts. So what types of polyploidy do we support in this module? The two major types are autopolyploidy, and this occurs simply when an organism gets more than 2 sets of chromosomes derived from the same species. Recombination can occur between any chromosome, so the number of clusters present is equal to the ploidy plus 1. The second type, which is a little bit less straightforward, is allopolyploidy. This is when an organism gets more than 2 haploid sets of chromosomes derived from different species. Recombination can only occur between chromosome pairs from the same origin, so basically we're only going to be looking for 3 clusters. This is an example of what you might see with an autotetraploid species. You'll notice that there are 5 distinct clusters, and they're all organized and clustered differently with different color codes. And you'll notice that the algorithm fits the number of clusters equal to the ploidy plus 1. So in this case, we are looking for 5 clusters. This is what you might see if you're looking at an allohexaploid species. So basically what you're doing here, the software is fitting 3 clusters, and the ploidy is basically only controlling the separation between these clusters. And you'll notice that we do have an inbred population option, which I'll explain later. Basically, you're telling the software not to look for heterozygotes. And this could be used, obviously, if you're working with an inbred population. So now that we've defined the types of polyploidy that are supported, let's talk a little bit about the algorithms. First and foremost is the Illumina developed Polygen Train algorithm. We're expecting you to use this one the most often. It's especially good for missing clusters. Say if you only have 1 or no data points in a certain cluster, it can actually infer that cluster for you. One of the options, which we'll also discuss later, is that you can model the allele frequency. This basically uses the minor allele frequency and Hardy-Weinberg equilibrium calculations to determine the likelihood of a cluster. The second algorithm that we offer is called DBSCAN. It's an acronym for Density-Based Spatial Clustering of Applications with Noise. Basically, this is a secondary option that you can use when Polygen Train does not yield the results that you want. It's especially good for clusters that are irregularly shaped or not distributed symmetrically. For instance, if you have a cluster that's significantly, say your pure homozygote clusters are significantly off-axis. This would be a better model to use because Polygen Train is actually looking for the clusters to be relatively symmetrically distributed. So there's 2 types of clustering options that are offered in this module. There's the general clustering options, which are universal to the entire project. One of these is setting a clustering intensity threshold. Basically, this is a normalized R value that removes low-intensity samples to allow you to optimize cluster identification. The second option is the inbred population. So if you know that you're working with an inbred population that's going to be lower diversity, you can tick this box, and this will basically ignore the possibility of a heterozygote cluster. And this would be especially useful with allopolyploidy. So in addition to the general clustering options, there are also algorithm-specific clustering options. In DBSCAN, one of those is cluster distance. And what you want to think of this as is essentially cluster radius. So if you increase this factor, the software is going to be looking for larger, less dense clusters. If you decrease it, it's looking for smaller, more dense clusters. And basically it will find more clusters the more you decrease this factor. You can also specify the minimum number of points in a cluster, but it's important to note with DBSCAN that you have to have more than one data point per cluster, otherwise it cannot actually designate a cluster. As for Polygen Train, there's only one algorithm-specific clustering option, and that's the model allele frequency option, which basically looks for a genotype distribution based on minor allele frequency and Hardy-Weinberg equilibrium calculations. As for clustering Snips, you will notice that it's much more of a manual process. You can select to cluster all Snips when you're creating the project, and when you do that, it will use the Polygen Train algorithm by default. Now, if you go through and you decide you want to cluster some Snips using DBSCAN, or you want to change some of the options and recluster, you can do that. But if you cluster up front during project creation, you will use Polygen Train for all Snips. So once you do that, then you can go in and right-click on any SNP in the full data table or SNP table, select Cluster Selected SNP As, and select Polygen Train or DBSCAN, depending on what you want to do. As for editing clusters, the process is very similar to the GenomeStudio Genotyping Module. You can click and drag the edge of the oval to change the size of the centroid, or you can click and drag the center of the centroids to change its location. And you can also delete or create clusters manually. In addition to that, you can also manually edit the cluster genotypes. If you decide that you don't like the way certain samples are called, you can manually select those samples and choose to include them in a specific genotype, depending on what you think is most appropriate. As for the middle of the process, this is obviously where you're going to be spending the most time. In general, we recommend following our standard Infinium genotyping workflow. First and foremost, you want to exclude poorly performing samples, and you can use the standard metrics such as call rate, score to do that. And in general, what you want to be looking for is any drop-off in the metrics. So say if you see a shoulder where call rate decreases significantly, that might be a good clue that anything below that are poorly performing samples. After you have only well-performing samples in the project, you'll want to zero any poorly performing SNPs. And just like in the genotyping module, you can use call frequency or cluster separation or other metrics to do this. Once you've zeroed your poorly performing SNPs, then you might want to go through and edit or optimize the clustering for the remaining SNPs with the remaining samples. Once you're happy with everything, you can export the cluster file, the .egtp, for future use. All QC thresholds and cutoffs are 100% customer determined. We don't have specifications for custom products, at least not in terms of call rate or other common metrics. So it is up to you to determine these thresholds. For those of you who'd like to learn a little bit more about the basics of genotyping and clustering, we do have a recorded webinar specifically on that, and I will show that on the resources page at the end of this presentation. Finally, after you've finished all of your analysis and you've exported your cluster file for future use, you can also generate final reports very similar to the way you do it in the genotyping project. You can use all of the standard allele and strand designations that we typically provide. So you can use allele forward, allele plus, AB, or top. So just a couple of reminders. It may be tempting to think, since you're working with polyploidy, that top, bottom strand, and AB allele no longer apply. They do still apply. Infinium is a biallelic assay, so we're still only looking for 2 nucleotides at a time. Just with polyploidy, we're looking for more at the same time. So you still have a top-bottom strand, double-stranded DNA, and you still have an AB allele, so to speak. So, for instance, if you're looking at an autotetraploid species, such as potato, the genotypes would simply be all possible combinations taken 4 at a time of A and B. And if it's a TG variant and you want the nucleotide format, it's just all possible combinations of T and G. It's also important to note that if you are an XT or otherwise high throughput customer, you'll want to use the GenomeStudio Polyploid Genotyping Module mainly to generate your cluster files. And then once you've done that, you'll want to use that cluster file and automate it with either autoconvert in Beeline or autocall if you have a LIMS system. So these are just a couple of resources that you can consult with as you're going through and learning how to do polyploid genotyping. The GenomeStudio support page, of course, is where you download the software. The Beeline support page, where you can download Beeline if you're interested in automation. We have a number of different array recorded webinars. I listed 3 that might be of interest. Our top, bottom strand, and AB allele technical note, and of course our array online training. Okay, so those are all the slides I have. Now I actually want to take you into GenomeStudio and do a quick demonstration of what you can expect to see. Okay, so this is a GenomeStudio Polyploid Genotyping project. This is actually a potato, and you'll notice first and foremost that instead of having 3 distinct clusters, we now have 5 clusters. Potato is a tetraploid organism, so we expect to see 5 clusters. The first thing I'd like to bring your attention to are the clustering options that we discussed. You access those by clicking the Tools menu, then going to Clustering Options. So as we discussed, there are high-level genotyping or polyploid genotyping options for clustering, and there are also algorithm-specific clustering options. For the general options, you can do a clustering intensity threshold. Right now I have it set to 0, but say you wanted to filter out all samples that were less than 0.1. You simply select that, click OK, and then recluster your selected SNP. Sorry about that. You have to make sure that you select all samples. So now you'll see that below 0.1, all of these samples have been excluded from the analysis. Say you decide that's not viable and you just want to reset it back to 0. Just change that back to 0 and recluster. So now everything is included again. So now, say that you're not happy with the way this particular SNP is being clustered. you can always go back and change the algorithm to DBSCAN. So you go to right-click, cluster selected SNP as DBSCAN. And you will notice that, of course, the clustering is much different now, and it's basically not finding nearly as many clusters. So what you can do in order to alleviate that, you can change the cluster distance so that it actually looks for larger clusters. So instead of 0.1, let's try doing 0.12 and see what happens. So now the clusters are actually, it's finding much larger clusters because we increase the cluster distance. Obviously, we're looking for 5 distinct clusters, so this probably is not a good cluster distance for this particular SNP. So let's try changing it back to 0.08. So again, it's finding a lot more clusters, but it still does not appear to be optimized. So maybe DBSCAN is not the best algorithm for this particular SNP. So with that said, let's go back to Polygen Train. So this obviously looks a lot more reasonable and probably what you would expect. So going back to kind of the overall Infinium workflow and data analysis, the first thing you would want to do is have a look at your major sample metrics. So first you want to eliminate poorly performing samples. One way you can do that is by simply sorting the call rate and then doing a line plot of call rate. Now, it doesn't really show it in this project, but many times you can see kind of a drop-off, a shoulder, where samples begin kind of sharply decreasing in call rate. That might be a good indication that samples below that threshold are poorly performing and need to be removed from the analysis. So once you've either used call rate or score or another metric to eliminate poorly performing samples, then you would actually want to go in and eliminate poorly performing SNPs. Generally, the easiest way to do this is within the SNP table, and you can sort, say, by call frequency. This way, you have the worst performing SNPs at the top, and you can choose either to eliminate these, zero them, or you can try to adjust the clustering to something that's more suitable. So after you've done that, you've eliminated your poorly performing samples, you eliminated poorly performing SNPs. At that point, say everything is fine, then you'll want to export your cluster positions. You do that simply in the File menu, Export Cluster Positions, and you can do for all selected SNPs, for all SNPs, or for certain SNPs, depending on your need. You might want to output just a subset of your SNPs and then just use that for another purpose. And just briefly, we can have a look at the controls dashboard. You'll see that since this is actually an XT product, the controls do look a bit different. And you'll also notice that only the sample-independent controls are responding. That's because the sample-dependent controls, in other words, the non-polymorphic controls, are specific to human sequence. So you should not see response from those. But you will see the sample-independent controls. So the staining, the extension, target removal, and hybridization. And those can give you a good idea of how your processing went in the lab. Okay, so that concludes the software demonstration.